All right, this is part two of our video series on movement, and we talked in the previous video about passive movement, passive diffusion, active movement that requires energy but goes upstream, right? So primary active transport, secondary active transport. Now what we want to do is see if we can put all of those together to move the things that we're about to eat, right? We see that Frosty's fries, dipping the fries in the Frosty and the hamburger there. We want to take all of that stuff and we want to move it across this enterocyte, the cell in the gut, into the blood. So on this side of the cell, we've got the blood. On this side of the cell, we've got the gut. We just ate all of this stuff, right? This is going down the mouth and into the gut, and we want to move it. So one of the first things up on the list is glucose. Let's think about the options that we have to move that glucose across the cell. Our options are passive or active. Which would you choose? Passive is free. What are its limitations? Active costs money. So what could we do? Well, let's say we tried passive for just a minute. How much could we move of glucose if it was by passive transport? Remember, passive movement is all based on gradients. So let's say we have a scenario. I'm just going to use a random number. We have 100. We have 100 glucose molecules, and we want to move that. So how much could we move? 100% of it? Well, what happens when we move half of it? So instead of 100, now I've got 50 out here, or in the cell, and I've got 50 here. I've moved 50% of it, but by the time I move 50% of it, what's happened? I've lost my gradient. Now there's no desire to have a net movement. So now what's coming in is going to be the same essentially as what's going out. This doesn't work because if I leave 50 molecules of glucose in my gut, where's it going to end up? It's going to end up in the large intestine where my gut flora eats it, has a party. They love this stuff. The next thing I know, I'm bloated. I've got diarrhea. I'm not very happy at all. That doesn't work. I've got to move 100% of my glucose. Now, to complicate factors, I maintain over in my blood glucose levels between, well, we'll just up to, for an average non-diabetic individual, 100 milligrams per deciliter of glucose in the blood. That's my blood glucose level. And with that, I have a significant wall to overcome as I try and move this glucose. So I hope you can appreciate now that moving glucose passively isn't really an option. We need active transport. Okay. So what do? Well, I could use primary active transport. I could use secondary active transport. The problem with primary active transport is every glucose I would move would require ATP. I don't know that that's the best way to use ATP. It would require a lot of ATP just to move the glucose. And the glucose is coming in to make ATP. So that doesn't really work. Is there a better, more efficient way to move all of this glucose? And the answer is yes, secondary active transport. So let's set this up and see what we can figure out. Of course, for secondary active transport, what do I need? I need a gradient. If we look over here on the basolateral surface of the cell, I've got ATP running my sodium-potassium pump. The purpose of primary active transport is to establish gradients. And we can see the two different gradients that are established by the sodium-potassium pump. If we do sodium, sodium is high on the outside and low on the inside, right? We see that because we're pumping 3 sodium out every time this runs. Now, on the flip side, we're also pumping 2 potassium in. So we see another gradient, but this gradient's inverse, where we have high potassium on the inside and low potassium on the outside. But for this purpose, what we're really interested in is sodium. Because sodium really wants to come inside the cell because of its concentration gradient. Also because of its electrical gradient. We'll talk about that at another time. regardless, sodium really, really wants to come inside the cell. So that's why salt and sugar go so well together. That's why people like dipping their salty French fries into a Frosty, because in order to move the glucose, I need to eat salt. As salt comes into the gut, salt wants to get in the cell because the ATP pump on the other side is pumping all of this sodium out. I can use the strength of the sodium gradient to move glucose. In other words, I say, sodium, I know you want to come in the cell. I'll let you come in as long as you pull glucose with you. This is secondary active transport. Glucose going against its gradient because sodium is pulling it along. And because we see these two arrows moving in the same direction, this is an example of symport. We also see that same sodium gradient moving amino acids, another symporter here. Let's look at this next one though, right? We've got oligopeptides, proteins essentially being broken down, and we need to move those. It is an example of co-transport. They're both coming in the same direction, so this is another symporter. But what's driving this? Here, it's being driven by a proton gradient. Well, how do we make the proton gradient? What I want to show to you is that all of this stuff can be traced back to the sodium-potassium pump. So to visualize that, let's also use sodium here to run an antiport exchange with protons, with H+, right? We see sodium going in, down its concentration gradient, and it's pushing protons out against its concentration gradient. This is how H+ gets high out here because of this sodium. And again, the sodium is running because of the sodium-potassium pump. Now, what does that do? Well, we've talked about the acid-base buffer system, the bicarb buffer system. If we're removing sodium, this is going to be the insult. If we're removing H+, this is going to be the insult to the equation inside the cell. H+ goes down because I'm pumping it out with the sodium gradient. As H+ goes down, that pulls the reaction to the right, leading to a decrease in CO2 and an increase in bicarb. As bicarb levels go up, that creates a gradient for bicarb. So now we can run this antiport exchange here using the bicarb gradient, and this is how we're moving chloride against its gradient into the cell. Of course, with the decrease in CO2, we set up a gradient for carbon dioxide. If you remember, CO2 fits the definition of small, nonpolar, non-charged, lipophilic, and I guess, of course, the CO2 is going to be over here in the blood, so we'll draw it over there. Now I have a gradient for CO2, and we see an example of simple diffusion as CO2 simply goes in to replace the CO2 that was converted into bicarb. So this is an example of simple diffusion. Now, we do have a couple of examples of facilitated diffusion. We see potassium here. We know that the gradient for potassium is high on the inside thanks to the sodium-potassium pump, and then we have a leak channel open. Potassium just turns around and leaves. This is important for setting up resting membrane potential, which is something that we'll talk about shortly in a new video. The other example of facilitated diffusion is fructose. Fructose is like glucose in the sense that it's a sugar, but it's unlike glucose in the sense that we don't have a big wall to climb up. Remember, glucose is in the blood. We showed this over here at 100 milligrams per deciliter. That's the wall that we had to climb up. That's why we needed active transport for glucose. But for fructose, the amount of fructose over here in the blood is essentially zero. And so the entire gradient for fructose, if I eat it, is high to low all the way across the cell. And there's not a whole lot of fructose to deal with anyway. And so fructose can move in this direction, high to low, into the blood because there's zero in the blood without significant problems. Hopefully this now gives you an example real life example of how all of these different dynamics work together. Primary active transport to create gradients, secondary active transport, usually using sodium somewhere because that's such a strong electrochemical gradient, to move all of these other things to set up additional gradients. And then we also see facilitated diffusion and simple diffusion in play as we move all of the things that we just ate, from protein and carbs and salt, all of these things across this gut enterocyte into the blood.